All right. Let's see if I can get my pens here. Mini online lecture is to help you transition from meiosis to genetics. It's also so you don't forget my lovely voice over Thanksgiving break. And so at some point you will get the secret password that you will type into the pre-class 13 quiz for full credit. All right, here we go. Excuse me. And of course, as soon as break started, I got sick. Isn't that how it always goes? I hope you all are doing well. We're going to cover some of these concepts about alleles and genes, the law of segregation, and the law of independent assortment. Okay, so genetics is talking about inheritance. So this is the passing down of traits, which is really DNA information from parents to offspring. So everybody technically has a biological mom and a biological dad, and you inherit half of your genetic information from mom via the egg, and half of your genetic information from dad via sperm. Okay. Remember when we talked about meiosis that egg and sperm are haploid, right? They have half the amount of genetic information. This is a little thick. And remember that people are diploid, which means you have chromosomes in pair, one from mom, one from dad. That means you also have 2 copies of every gene. 1 copy from your mom, 1 copy from your dad. So let's define a few terms. Okay. We've been talking about the term gene. So a gene is a region of DNA on a chromosome that gets transcribed right? So you make mRNA, and that mRNA will eventually get translated into protein. And these proteins are really what gives you the traits. So a trait is your physical characteristics. I don't know why it's doing that. Okay. We are also going to use the term phenotype to talk about someone's physical characteristics. Okay. So in this case, we have the example of hair color, right? Lots of different hair color shades. So there can be a gene for hair color. The allele is a version of a gene. So we're talking each allele. Ugh, I don't like this. I think this one's still thick. Each allele, I'm going to use a different color. Each allele has a different DNA sequence. Okay, so you inherit 2 genes for hair color, one from your mom, one from your dad. maybe you inherit the blonde allele, and maybe you inherit the brown allele. These are your, I don't know, how can an electronic pen, do you see how it's like this? How does an electronic pen leak? Okay, I tell you. All right, so let's recap. You have 2 versions of each gene. Okay. Wow, I don't know what's going on. One from mom, one from dad. That means you have 2 alleles. The way those alleles interact is going to make your trait, your physical characteristic, what we actually see. And that's going to really be what we talk about in classes. How do you interpret all this gene and allele information to get phenotype? So again, a gene is a small region on a chromosome. It gets transcribed. It makes into a protein. That's going to give you some physical characteristics. So, genotype versus phenotype. Genotype is your genetics, your DNA. Ugh, this is so weird. Wait a second. 1, 2, 3. Okay. Sorry. Phenotype is your physical characteristics. What you see. Are you tall? Do you have brown hair? Do you have blue eyes? Do you have dark skin? What do you physically look like? Okay. So you could have the phenotype of blue eyes. Then we look at your genetics, your DNA, and see what your genotype is. You could have the phenotype of brown eyes, and you might have multiple, or there might be multiple genotypes that can cause that phenotype. That's what we're going to talk about in class. This cool thing is called a Punnett square. A Punnett square is where you take the sperm and the egg and you determine possible offspring based on genotype, and you interpret it to phenotype. Oh my gosh, this is just driving me crazy. Why is it doing this? Hold on just a second. Let's see if I can. No. I don't think this is going to help me. Sorry, I just can't stand that. How it's like, whatever. Alright, sorry. You know me and technology. Alright, what I want you to notice, excuse me, is that the sperm and the egg, they only have 1 copy of every gene. So when we talk about a gene, I guess I need to back up. Let me add, whoops. Let me add 1 slide in here. Okay. Your genes are going to be represented by 1 letter. Okay. So you could have a gene represented by the letter A or the letter B. Okay. The alleles are going to be represented by either big A or little a, big B or little b. So 1 gene, same letters, but you can have capital or lowercase. And we're going to talk about dominant and recessive in class. Okay. What I want you to understand is that a diploid organism has 2 copies of each gene, right? So they could have big A and big A, or they could have big A and little a. They could have little a and little a, right? That's one gene, 2 alleles. Okay, so let's look at our example here. How many genes are we looking at in this Punnett square? Since there are 2 letters, we're looking at 2 genes. Okay, we're looking at the S gene and the B gene. And do you notice how every single one of these cats has 2 copies for each gene? So it has a big S, or big S, or little s. But it has 2 copies. It has 2 alleles for every gene because it's diploid. Okay. In contrast, the egg and sperm are haploid. So they're only going to have 1 allele for each gene. They can have little s and little b, or big S or big B, right? But they have to have 1 gene, or 1 allele, sorry, for each gene. And when you combine them, egg and sperm, you're back to diploid. So haploid equals diploid. And where we're going with this is to connect meiosis that you've learned about to genetics. All right, so again, this just reiterates what we've been talking about. Okay. Diploid organisms have 2 copies of each gene, which means they have 2 alleles. And the alleles can be the same, they can be different. So in this example, the gene is for flower color. The alleles are purple and white. Oh, this is really driving me crazy. I don't know why the pen's doing this. Okay. So, you have 2 copies of the flower color. 2 copies of the flower color gene. In this case, the organism has 2 different alleles, 1 purple, 1 white. So let's connect this to meiosis. So let's say you are a diploid organism. And you have a chromosome. 1 has the big A allele, 1 has the little a allele. So it's the A gene, and it could be the A gene, right? And the alleles, the variations, are big A or little a. So in your gametes, through meiosis, which we will go through, your gametes are going to have either the big A or the little a. Your gametes are going to be haploid. Okay? So you go from a diploid organism to haploid gametes. The first law we're going to talk about of Mendel is the law of segregation. And the law of segregation is what you have been doing in meiosis. And that just means that chromosome pairs separate so that each gamete receives 1 allele for each trait. Okay? You were already doing this, we just hadn't put any gene allele information in with meiosis. Okay? So I want you to see that you have a chromosome that has an A and an M and a B gene, and that every single gamete has, what's the M? Oh, sorry. You have an A and a B gene. M standing for maternal, the plus is for paternal, sorry. And every gamete has an A and a B, a chromosome, sorry. And, okay, let's start again. The diploid cell here, this would be your germline cell, has 2 chromosomes, right? This is your homologous pair. It has the genes A and B on it. We know through meiosis we duplicate those chromosomes, we allow crossing over to happen, we split the homologous pairs, we split the sisters. In the gametes, each gamete has a chromosome with an A and a B gene on it. You have different combinations, and that's what we were talking about in meiosis and crossing over. So, chromosomes separate so that each gamete receives 1 allele for each trait. What about when this gets complicated? We have multiple chromosomes, multiple genes. So what I want you to see here is you have 1 homologous pair with the A and the B genes, and another homologous pair with the C and the D genes, which means each gamete has to have one or the other AB chromosome and one or the other CD chromosome. So let's follow this through meiosis. It's telling my Wi-Fi. Okay, we're just going to do 1 crossing over per homologous pair. So this is prophase 1 of meiosis. We've got some crossing over happening between the tips of these chromosomes. And so you get genetic exchange, and they're going to line up, right, in metaphase 1, and you're going to basically split them down the middle. So that at the end of meiosis 1, you have split the pairs, the homologous pairs, right? Each new cell still has B chromosomes, AB chromosomes, and CD chromosomes. We've had a little bit of genetic exchange. These line up for metaphase 2, and remember now we're going to, excuse me, I can draw these, split the sisters, and after meiosis 2, you end up with 4 gametes, non-identical, and we've been drawing this with our colors, and now all I've added is some gene information, allele information to these chromosomes. Okay? So each are genetically unique. Each gamete has an AB chromosome and a CD chromosome. They're all haploid. They only have 1 copy of each chromosome. So what we're going to do now is what you're going to see as we talk about genetics, and we just list the genotypes. So the genotype for this gamete, we just follow the letters A, B, C, D. And the reason I underline C is when you use uppercase and lowercase that look the same, sometimes you get confused. Did I write that big or little? So mark it somehow. The genotype, the genetics for this second gamete is little a, big B, little c, big D. The genotype for the third gamete, big A, little b, big C, little d. And the genotype for the fourth gamete is little a, little b, little c, little d. So these are all different. They are all genetically unique. And if you were drawing this on your own, you may have drawn it slightly different. And that's okay, because the law of independent assortment says any, in this case we'll talk about the AB chromosome. I know these say Y and R, but going to our back figure, can segregate, or I don't know why this does this, move with any CD chromosome. So you're going to have more combinations than what we just drew, and we're going to look at that in just a second. So the law of independent assortment means any chromosome, as long as you've got one of each, can go with any other chromosome. So they're looking at a genotype of the parent, big Y, big R, little r. And they don't have multiple genes on a chromosome, so they're not worried about crossing over here. But what it's showing you is the chromosomes with the lowercase alleles can go together, or they can go with big alleles, right? There's no rule that says the way you line it up, let's see, where are we? Up here, or even all the way up here, you could have these switched. So you could have the pink on the other side and the green on the other side, right? Or crossing over might happen between the outer pieces. Okay. You can get all possible allele combinations. That's the law of independent assortment. Any chromosome can go with any other one as long as you have one of each. So you can imagine with 23 pairs of chromosomes, you have lots and lots of combinations. With just our little group, so our parents, if you looked way back here, the genotype of the parent of the diploid was big A, big B, little b. You always write the allele pairs together. Big C, big D, little d. So we had 4 genes, each with 2 alleles. When we put these together, there are 16 different possible gametes. So let's just go through these to look. You could have, oh, why is this doing this again? I was like shaking up my battery and it stopped. I don't know what's going on. I need to ask Santa Claus for a new Surface or iPad or something. So, big A, big B, little c, big D. That's one allele combination for this gamete. Little a, big B, big C, little d. big A, little b, big C, big D. And I will not read all of these to you because you're going to get tired of hearing me. But if you went through and did every possible combination, and this is what we will do in class, yay. Now, how come that one's the same? Interesting. 2, 4, 8, 6. Yeah, alright, we'll quit now because I didn't quite get these all mixed up. But anyways, trust me, there's 16 different possibilities. Alright, so the summary. What you should have learned from this video. When I say the word genotype, you think genetics, you think DNA. Each gene is represented by a letter. The allele is represented by either a capital or a lowercase. But it has to be the same letter. So one letter, one gene. All the organisms we're going to talk about are diploid, which means they have 2 alleles. All the gametes that we're going to talk about are haploid. They have 1 allele per gene. And when we say phenotype, we're talking about the physical characteristics. Due to allele combinations. Okay, we will talk more about how we determine phenotypes, in class. So, a couple clickers to try out and see how you're doing with this. Does that work? Okay. Which of the following is a correct representation of a human genotype for one gene, the hairy back gene? So you might want to pause this, pick your answer. The answer is C. Okay. For the genotype, you have to have 2 alleles of this for this human, and they have to be the same letter. So this person has 2 capital letters for the hairy back gene. Hairy back description would be phenotype. Okay. A single letter would talk about a gamete, and an H and B would talk about a gamete with 2 genes, because each letter represents a different gene. Okay, next one. Which of the following is the correct representation of a human genotype for 2 genes, the hairy back gene and the blue eyes gene? We'll go ahead and pause. And the answer has to be B. Your genotype has to have 2 alleles per gene. So HB or HB would represent 2 genes, but in a gamete. HH or HH would represent 2 alleles, but for only 1 gene. Okay, so since we have the H gene and the B gene, we need to have 2 alleles for each gene. Which of the following is the correct gametes for the following genotype? So this is the parent. And which kind excuse me, of gametes would they make? And this is where I'm giving you code word Mendel. This is what you're going to type in for pre-class 13 quiz. Mendel, the father of genetics. Okay, let's go back to this one. Gametes are only going to have one, so you could make either an H gamete or a little h gamete, but because these have two, these are diploid, they cannot be gametes. All right, one more. Which of the following is the correct gametes from the following genotype? So we've got two genes. Pause. Okay, this is tricky, and this is going to be how you figure out genetics. The correct answer is this. So what I'm going to teach you is that you can have H with big B, H with big B, that's the same, right? Or you could have little h with big B, little h with big B, that's the same. So the unique gametes are big H, big B, little h, big B. I'm going to show you in class how to figure this out. What I want you to see is the reason this answer is incorrect is because there's no little b above, right? This person doesn't have a little b allele. This is diploid. Gametes must have one of each. And these don't have enough genetic information, right? You got to have one of each. So again, last reminder, code word mental. Make sure you spell it right. Go put it in pre-quiz 13. I will see you on Monday the 26th. Happy Thanksgiving.